This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded on September 13th, 2016. I'm Kevin Bonham, and it's been a while since we recorded, but I'm joined this evening by Matt Woodruff.
How's it going, everyone?
And no Kate today. She is busy rallying the people against the man.
Yeah, she's doing a pretty good job, actually. Seems like.
I've heard good things. The grad student union at Harvard is organizing. I recently found out that as part of the UCSD postdocs that I am a union member.
Oh yeah.
And they are organizing strike action. I didn't know postdocs could strike. That strikes me as almost a contradiction.
Theoretically, they should be able to, right? But would that just mean they're working like 30 hours a week?
Yeah, that was my impression, because they're not going to stop taking care of their mouse colony, right? They're not going to stop doing experiments that are the thing they need to get a job. It strikes me as like they're going to strike and basically not get paid for continuing to do the work. It doesn't make any sense.
Maybe it's like a strike
I authorized the strike action.
Yeah, like a strike in spirit. Maybe you could like not go to a lab meeting or something. Mm-hmm. And then write a really apologetic email afterwards about why you weren't in lab meeting.
Yeah, I was planning to send my PI a note that says, can you just tell all your colleagues how annoyed you are that I'm not working?
Right.
I continue to work.
It's the optics that I'm after here. It's not actually a reduction in work. Also, please help me publish my paper.
Right. And write me a letter of recommendation for my next job, and etc.
Yeah.
Yeah, there are some fucked up incentives being a postdoc.
Yeah, I mean, the thing is that everyone is sort of everyone's out for themselves, right? Because it's so individualistic, it's your own career that gets hurt if ultimately you don't publish a paper. Right. So because there's no defined outlet, I think it just puts everybody in this really terrible position. But what's interesting about that is I think that there's just as much leverage in the other direction. As much as the postdoc strike sounds like a ridiculous idea, I have often wondered what would happen if everyone actually just put down their pipettes and walked out of a lab. Can you imagine a 20 postdoc lab just deciding, you know what, we're not going to come into work for 2 weeks? As much as the optics would be bad in the one direction, imagine all of the other PIs around a PI where 20 people just walked out of their lab. It feels like there would be a lot of backlash.
Yeah, I think you're right. It's just there's the free rider problem, right? Where you have the collective action problem, I guess they're related.
Yeah.
If the one postdoc that sticks around to continue to do their experiments, that postdoc is going to get whatever benefit, but not have to suffer the consequences. So,
right, but they can't. But the one person that sticks around isn't going to be more productive than they were beforehand. You know what I mean? So I just don't know. You can't make up the labor. So you can always get a scab in an auto worker environment, right? That will cross the picket line, do the same job for lesser pay. Mm-hmm. It just doesn't really exist because it feels like postdocs are already scabs. They are at some level,
they're paid
right?
So little money for such skilled work. Yeah,
right. And so obviously you can get people in the door that can pipette and that can work with mice, and pretty easy to find a replacement from the massive glut of PhDs that currently exist, but if everyone takes their lab notebooks with them, there's really no recourse from the high end of the lab.
Yeah, that seems true. Anyway, revolution aside, what are you drinking this evening, Matt?
So I'm being sort of lame, and I'm drinking water. Because I was tired, and I figured I would fall asleep while we were recording this.
So I'm also drinking water because I just got home from a bike ride. I literally, parked the bike and walked straight up here and started recording, but I am trying to not be lame, and I am drinking a rather interesting beer.
Oh yeah?
This is from a growler.
Okay. It's always better from a growler.
Yeah, I feel that way. I was up in Vermont for vacation last week, and on the way home, we stopped at a brewery that I really like called Hermit Thrush, which is in Brattleboro, Vermont.
Okay.
and they brew almost entirely with wild yeast.
That fits right
And
into your style, doesn't it?
oh yeah, and they have a lot of sort of weird, funky flavors, particularly sour flavors from Brettanomyces and Lactobacilli. Um, this brew is called the Sprucey Friar. They have a whole range of beers that are the same base beer, but that they steep in different things. So
Huh.
this one is steeped with spruce tips.
No, so it's like a
So it's got
gin
a little bit
beer.
of a sprucey flavor. They also have the Rosy Friar, and the Silly Friar is the base beer.
Hmm.
I
I,
feel like
al-
there's another one, but this is quite tasty, a bit sour, a little bit of a pine flavor. It's quite good.
I almost feel like you'd have to name the beer after you drink it for the first time if you're doing wilds ferments.
Hmm.
We reserve the right to name this beer later depending on what it tastes like that day.
Yeah, yeah, and it's going to be different every time because they're cultures. I am like super keen to get cultures from this brewery. And actually, one of the founders of the brewery went to school with my wife,
Hmm.
so I have a personal connection. I'm hoping to study their cultures because they're going to evolve over time, right? They're going to be like,
You'd imagine they'd have to.
yeah, they're going to be cycling them and reusing them over and over in multiple brews. So these things are all going to co-evolve, and I think it'd be a really fascinating microbial question.
Yeah, yeah. So speaking of evolution and natural selection and pressure, I
Nice
have good
transition
news.
there,
I
Matt.
have good news for you.
What is your good news?
While the Tasmanian devil population is plummeting rapidly, it's not plummeting quite as fast as everyone thought it was.
It hasn't hit the ground yet, which everybody
No.
was expecting.
Right. Yeah. So it turns out that it's been predicted for a while now. And I think that we've touched on this in previous episodes, but
We should say we're onto the paper now.
yeah,
Folks.
we're now no longer talking about beer in that smooth as silk transition. So, so Tasmanian devils for a while now have been dealing with this terrible disease. It's a facial cancer. It's called Devil Facial Tumor Disease, which may be the worst name for a disease that you could ever have.
Or greatest name for a disease ever?
I suppose that depends if you're taking the perspective of the disease.
Right.
So, so if you're into, sort of grotesque images of terrible diseases, the Wikipedia page for this does a pretty good job of showing what this disease is. But
Of
basically,
horrifying you and
yeah, it's
interrupting your
awful.
sleep.
It's awful. So essentially, it's a facial cancer, and it gets really out of control. And the problem with this facial cancer is that it is one of, I think, 3 known cancers. It's actually 4 now because they found a second devil facial tumor disease.
Right.
But
Unrelated.
right, unrelated, different primary source, but it's one of 4 diseases, 4 cancers that are known to be transmissible from organism to organism. And we've talked about this in the past. This is not something that generally happens. And the reason that it's something that doesn't generally happen is that there's a lot of immune diversity between individuals of a species. And the idea there, or the selection there, is that if you get some sort of disease that comes into the population,
We're talking about infectious disease now.
infectious disease, yep. Your immune system, person to person or devil to devil, should be sufficiently diverse so that even if the pathogen takes out 1 or 2 of you, you have enough diversity in the population in your ability to respond that most of you will survive and the species will continue.
Right. And we're talking here predominantly in terms of diversity, we're talking here about major histocompatibility complex genes. So this is, I think we've touched on this before. I think maybe even in our very first episode about transplantation,
Yeah.
we talked about MHC. And this is an allele where not only do we have gene duplication, so you have multiple alleles for MHC, both class 1 and class 2, you also have incredible allelic diversity, meaning that, at least in human populations, very few humans have identical MHC profiles because there are multiple alleles and each allele has a ton of diversity. So, as Matt was saying, the notion here is that in a given population, you're going to have enough diversity that microbes are going to not be able to just completely ravage an entire population. Now, I should say that there's some weird dynamics here because it's not really group selection. There are individual benefits of being different in a population. There's a really great book called The Red Queen that that basically posits that sexual reproduction evolved specifically for this purpose of combating microbes, and we see it in MHC diversity. But that's an aside. Sorry.
No, it's fine. So, yeah, I think it might be worth. I know that we've talked about the function of MHC a lot, but it might be worth just touching on what it does very briefly. So, we've talked about a couple different cell types. One of the cell types that we've talked a lot about in the immune system are T cells. And T cells are one of the major lymphocyte populations that are going to be responsible for fighting off pathogens. People probably know this idea of killer function. So the ability of a cell to identify another cell that's infected and directly kill it to wipe out whatever pathogen is inside. This function is largely mediated by the T cell population. And also T cells are really important for things like the generation of antibody responses. They help out macrophages, so they assist with innate immunity. They're really sort of the central lymphocyte as far as directing an ongoing immune response. And we've talked a lot in the past about how these T cells sort of know that they're specific for a pathogen. All right, so unlike B cells, which, you have a B cell and it bumps into a virus, and that B cell has a B cell receptor, and the receptor either fits the virus or it doesn't. That is, it's shaped and charged in such a way that it can interact with that virus effectively, or it doesn't, and that's how we get B cell specificity. T cells, on the other hand, don't see the intact virus. They see little snippets of the virus, and they see snippets of the virus presented on the outside of members of often the innate immune system, and we call these cells antigen-presenting cells. And their function is to take that virus, internalize it, break it up into little bitty pieces, and then present it on the outside of the cell. So when the virus is broken down, you need some sort of machinery in order to take those viral bits. In this case, we're talking about peptides, so the individual proteins in the virus are broken down. And those peptides are loaded onto a complex called MHC. And so when we're talking about MHC diversity, what we're really talking about is diversity in the protein that antigen-presenting cells are using in order to sort of show your T cells what's going on in your body.
I should say, if you want a longer description of this, you should check out episode 7, Hot Dog Buns, Hard Lines, and Floppy Bits.
That was my favorite title, I think, of any of the podcasts that we've done.
It's pretty great.
I really liked that title. So we have this huge amount of MHC diversity that is really important in making sure that when a particular virus or a particular bacteria comes in, at least some of us in our population have antigen-presenting cells that are capable of loading pieces of that virus or bacteria onto MHC and presenting effectively to T cells, right? And so, as you mentioned, we have a huge diversity in the human population. There are thousands and thousands of alleles that humans have. And so this is why tissue banking is so critical. It's why when somebody needs some sort of organ transplant, you go into the tissue donor list and you search through thousands upon thousands of donors. The reason you need to do that is because it's extraordinarily unlikely that my MHC are anything like your MHC. And
that's particularly important too because the MHC is the target of the T cell receptor, which is this major component of the immune system. And for reasons that it's probably not worth getting into right now, the way that T cells develop is specifically to recognize MHC. If you have different MHC alleles, you have this huge chance of triggering an immune response. This is why organs get rejected in transplantation, because the immune system is constantly scanning MHC. The T cell receptor compartment is constantly scanning MHC, And so other other types of proteins that are different can in principle cause organ rejection. You have what are called minor histocompatibility genes, but beyond ABO blood groups, which will cause immediate rejection for various reasons, I think we've discussed as well, mismatches in MHC are the principal cause of organ transplant rejection.
Yeah.
And also, incidentally, the reason that most tumors are not transmissible from individual to individual.
Exactly. So if you develop a tumor, so let's hope that that never happens, but let's say you were to generate a tumor and somehow I was exposed to that tumor, right? Your tumor would be expressing your own MHC alleles, and my T cells would be looking across every single cell to make sure that that MHC lines up. And as soon as my T cells recognize those MHC molecules and that they're different, my T cells will immediately reject that tumor. And so it's virtually impossible for you to give me a tumor, right? It is non-transmissible. It's as non-transmissible as if you were to cut off your arm and sew it onto my body. It would be rejected in exactly the same way.
I was actually, in the process of reading this paper and thinking about it, I was actually wondering if identical twins could have transmissible tumors. Like if one twin got a B-cell lymphoma or something,
Yeah.
and then those twins shared a needle or one had a blood transfusion, like could that be transmissible?
I mean,
It seems like it should be able to.
Yeah, you'd have to imagine that in principle it might be. Now,
And then the other question is if you are on immunosuppressive drugs because you've had a organ transplant,
yeah,
would you then be susceptible to transmissible tumors? And I did a little tiny bit of PubMed searching, and I didn't find any examples of this, but it seems like in principle, it should be possible.
it seems like you'd certainly have a higher chance. I mean, you talk about how tumors are evaluated in mice, and the first way to know, one of the first tests that you do on a tumor cell line is to put it into a mouse that has absolutely no immune system. Right. And then if the tumor grows out, then it is a viable tumor and it can be transmitted animal to animal. The second test that you can do is you can take that tumor and you can put it into a mouse that is genetically identical at the MHC, but that has a full complement of the immune system. And then that sort of gives you a sense of whether the tumor not only is capable of growing in the new host, but also whether it's capable of modulating the host's immune system. Right. And so I think that that's actually pretty close to your identical twin situation. The one caveat to that is that as tumors grow, they have a tendency to modify the host's immune system incrementally.
Right.
So I think it probably depends in your twin situation just how advanced that tumor is and how the tumor has evaded the immune system, because it's totally possible as well that that tumor, when it gets transplanted into the new twin with a totally fine immune system, the tumor just wouldn't have the bag of tricks yet to totally shut everything down. So that's my guess. Mm-hmm. Mm-hmm. In any case, Tasmanian devils have it pretty rough. And the reason for that seems to be that somewhere around 6,000 years ago, there was a massive bottlenecking at the MHC allele. And so while humans have thousands of alleles at the MHC, Tasmanian devils are very highly restricted. And this is something that people really worry about when they're talking about death spirals from genetic bottlenecking, right? So basically you get a highly homogeneous group of individuals that makes up the species. And the problem with that, of course, as we alluded to earlier, is if a pathogen comes in that's capable of killing off these devils, or these killing off these devils is strangely
quite
provocative. Yeah. But so if you get a pathogen that comes in that's capable of killing one Tasmanian devil, because that devil's immune system is so close to the one next to it, it's very likely that that pathogen will kill off the one next to it as well. And so as I mentioned, about 6,000 years ago, there seems to have been this huge bottlenecking event. And now we have a Tasmanian devil population that is largely homologous at the MHC.
Yes, and we should say this paper didn't describe what caused the population bottleneck. Tasmanian devils are super awesome, by the way. They're a large carnivorous marsupial, so they're kind of like, wolves or predatory cats, except they're marsupials, so they're genetically related to like kangaroos, and I'm not sure that they describe what the population bottleneck is. They're on Tasmania, which is a relatively small island. So it's possible the population bottleneck had to do with isolation on this island. It's also possible that there was some infectious disease that wiped out a whole bunch of individuals that didn't have a particular combination of MHC alleles for some reason. This is thought to have happened in some primate populations. You have what's called a selective sweep of non-resistant alleles. So go ahead.
No, I think you were doing just fine.
Okay. So I don't know if the reason for this particular bottleneck is known or if there are hypotheses, but those are sort of some of the possibilities. But the end result is that you have this very limited number of MHC alleles in the population, which means that the population is sort of possibly susceptible to an infectious tumor. The other thing about Tasmanian devils that sort of like the second hit for this particular population is it turns out that they are very social creatures, but the way that they express their sociability is by biting each other in the face.
Right. And really, it's like any emotion, too. It's not restricted to like a single thing that they do.
Yeah, it's not like I'm angry, so I'm going to bite you. It's like, oh, you're sexy, I'm going to bite you. Oh, you're in my territory, I'm going to bite you. Hello, bite you.
Right, exactly. Yeah, so they express their feelings through vicious biting of the face.
Yep. And so a tumor that is present on the face and is potentially transmissible because of this low MHC diversity, and it turns out has the ability to downregulate certain immune responses, as we're going to see, that tumor is then more likely to be transmissible. And because Tasmanian devils, whenever they hang out, which is often because they're a fairly social species, whenever they hang out, they bite each other on the face, and so there's lots of blood, as you might imagine, and so it turns out that this is how this tumor, and in fact these tumors, because as you said, there's now a second facial tumor disease with a different origin, which is just fucking unbelievable. That's how these tumors seem to have come about. And as you said at the beginning, have pretty severely decimated this population. I mean, it's sort of shocking that these animals are not extinct at this point.
Yeah. I mean, they've done several epidemiological studies, and essentially, well, first of all, the numbers, and I think you mentioned them a little bit earlier. Actually, why don't we at least introduce the paper here, because I feel like we're going to start drawing data. But the title of the paper is called "Rapid Evolutionary Response to a Transmissible Cancer in Tasmanian Devils." The first author on the paper is Brendan Epstein, and the last authors, Paul Hohenlohe.
Sounds
Is that
right.
how you would go with that? Paul Hohenlohe and Andrew Storfer.
And we'll put a link to this in the show notes, and this is an open access paper in Nature Communications, so everybody can go take a look. And it's pretty readable, I found.
Yeah, no, it's pretty good. It was, I think, it looks like accepted in July of 2016. It was just published at the end of August, so pretty recent here. But just some of the numbers in the introduction. So this facial cancer is virtually 100% fatal. We're not talking about a disease that leaves many organisms alive. So it's basically 100% fatal. And the result of that is that in localized areas, localized populations, they've seen more than 90% declines within some populations, and they've seen essentially an overall species decline of more than 80% in less than 20 years. So we're talking about an enormous selective pressure and enormous burden on the population. And they tried to get at that a little bit by figure one. And I was reading through this and I was really excited that a map was a figure of this. And it makes total sense that they would include a map. But basically, what they show is the extent of the disease or the disease front across the Tasmanian island. And so basically, all they're showing is that where it was first discovered in 1996, sort of in the northeast corner of the island, by 2000, it had expanded significantly. And now by 2015, there are very few places in Tasmania where the disease has not been seen now in local populations, which is really tragic if the cancer is basically 100% fatal. And so,
Yeah, and this is a population that, as we said, is already bottlenecked. So there's already a very small amount of diversity in this population, and this is just contributing to even more destruction.
right. And so when the disease was first discovered and as it was developing, people basically did some epidemiological studies and started predicting how long this species is going to last, at least in the wild. It turns out that there are two sort of backup populations that they're keeping disease-free off the islands just in case they have to repopulate. But again, you worry about things like genetic bottlenecking, and you always wonder when you release them back out into the wild how well they survive. So it's not a great backup plan, but at least it's something. But they predicted, as they were sort of developing an understanding of this disease, that the entire population would be gone within 35 years, essentially. So total extinction of a species within 35 years is pretty dramatic, I would say. But fortunately, or at least fortunately for now, the whole purpose of this paper is that authors have gone into some of the populations that epidemiologically should at this point be extinct. And it turns out they're not yet. Now, they're mostly extinct, don't get me wrong. I mean, we're talking about a very small number of individuals out in the wild, and it could be, hopefully not, but it could be that these animals will eventually face extinction out in the wild, but they are finding individuals persisting in areas that have been dealing with this facial tumor for a long time. And so the basic premise of the paper is authors had already been pulling samples from these populations prior to the spread of the disease. So these are Tasmanian devil scientists, biologists. And so they've been collecting samples for a long time before and now after the disease. And they started to realize that there are certain genetic traits that are seemingly under incredible selective pressure. And they're starting to hypothesize, at least, that some of these traits that have emerged in these populations are basically responsible for the survival of these local populations much longer than was anticipated based on the initial death rate.
Right. And so I'm not sure that it's worth going into description of genome-wide association studies because it's a somewhat complicated morass and prone to a lot of misconceptions. But basically, the idea here is that you look at the population before the disease was prevalent, and you look at the populations that are surviving in the face of this disease, and you can... Nice, that was unintentional. And you can basically use statistical methods to map regions of the genome that seem to be associated with survival. In other words, like imagine you have region A on some chromosome, and there are two different versions of this region in the population. And prior to the disease, version 1 is present at like 40%, and version 2 is present at 60%. But then you look at the populations after the disease, and version 2 has jumped up to 90%. You might imagine that some gene in the vicinity of region 2 has some protective effect, because most of the individuals, after the disease has come through, tend to have version 2. Right. So this is just a generic example, and you can do this over the entire genome. You're looking at something called single nucleotide polymorphisms to get a sense of which genes are, or which regions of the genome are linked to survival. But I think one of the major misconceptions, and the thing that to pay attention to, is the fact that the single nucleotide polymorphism, the specific mutation that's being looked at as the marker, is not necessarily causal. It is just nearby the thing that is causal. So you can't necessarily link it to a specific gene, but you can link it to a region, and then you can look at the genes around that region and say, do any of these look like something that might play a role? So figure 2 is essentially showing you all of the positions for these single nucleotide polymorphisms that seem to be associated with increased survival in these post-disease populations.
Right.
And go ahead.
And it might be worth mentioning that you can do this in that sort of, what was it that you referred to it as, a sweep, a genetic sweep?
Mm-hmm.
Right. So in a case where, in the case where a pathogen enters into a population and it decimates the population, and then you go back and you look for what's been enriched. Kevin mentioned before that oftentimes what will happen is you'll get a restriction, and you'll get a restriction specifically in the MHC gene, which is what we've been talking about, right? So oftentimes when these sorts of pathogens come through and there's a huge selective pressure, the diversity between individuals, specifically at the MHC, is often the cause of survival. Unfortunately for Tasmanian devils, there isn't a ton of MHC diversity. Right, so the genes that we're looking at here, while you generally expect MHC alleles to be really high on sort of the protective level of an individual, or the protective signature of an individual, in Tasmanian devils, they don't have that resource to draw on. So instead, you're going to be looking at genes that, I don't know if you have different thoughts, but seem to be sort of vaguely related to the immune system in this case. And so you're just sort of grasping at straws a little bit. But it does seem like there is a selective pressure, and they are at least able to come up with genes in these regions that seem to be selected for, that may or may not be related to immune function.
Yeah, and I think it's evolution is really hard, and computational evolution is pretty tough, and it's easy to convince yourself that things that are enriched because they are causal, when in fact, oftentimes, because of the random nature of evolution and selection, sometimes you get things that are enriched in surviving populations purely by chance. Like, it just happens that one population is protected, and that enriches for something that is not actually causal. So I think there's a certain amount of judgment that has to go into looking at GWAS studies. You have to say, does this seem like a plausible link? And also, the genes that they pull out as being potential candidates are just that. They're just potential. And I think it's going to take a lot more work to identify if any of these genes are actually causal. But the genes they pull out that seem to be genetically linked to single nucleotide polymorphisms that are enriched in these surviving devil populations do seem to be related to cancer and immune function, which kind of makes sense. And I'm not sure if they pulled these genes because they make sense, or if these were just the genes that were pulled out and they happen to make sense. And I don't know if it's worth actually going into the genes they pulled out. Again, the paper is open access. You can go check them out if you're interested. But basically, they found that there are a number of genes involved in regulating the immune system, in immune detection, or in the cell cycle, which is, tumors, are typically genes that are related to cancer. So that kind of makes sense. And it turns out that these genes, certain alleles of these genes, are under increased selective pressure. Now, how these alleles are different from the ones that make the devils susceptible to cancer is totally unexplored, unknown, or whether they're even causal in the survival is also completely unexplored.
Or whether the
But there's
SNP even causes a change in the function of the gene, right?
yeah.
So,
So it's these are genes that are near the SNP. They are not necessarily genes that the SNP is in. That's one of the weird things about GWAS is that genes that are nearby the SNP, even if the SNP is in a protein coding gene, it need not be that gene that is associated with the SNP that is
right.
causal for the selection, because anything that is nearby is going to be genetically linked.
Right. So
Um,
we don't know. I guess what I'm saying is we don't even know if these genes are functioning differently between pre and post face tumor devils. We just know that they're in the right region. They could be related,
right.
right? It could very well be that so they've got 6 genes from chromosome 3 here, and at least 5 of them they can stick on some sort of immune function to.
Right.
It could very well be that the only gene that actually matters in that is, let's say, their cell adhesion retinal degeneration gene, and all the rest are functioning exactly the same. And it's
Right. And,
actually,
and also, it
go
may
ahead.
not even change the function. It may only change the regulation, like how much this gene is expressed or something.
Right, exactly. So, yeah, there's it's I guess 2 things that are good to know here. One is that there does seem to be some sort of selective pressure on the genome, and the second, obviously, is that selective pressure is resulting in these populations surviving longer, I think, than epidemiologists thought they would.
Right.
So, what you would hope, certainly, is that this would be some sort of stable mutation that wouldn't necessarily affect the long term survival of the population and somehow makes them resistant to these facial tumors. Although it is concerning that there is now a second facial tumor in the population, and it would be
Right.
interesting to know, of course, whether these same genes are being selected for, or whether that second facial tumor is just going to come through and wipe out all of these mutants, and that'll be the end of it.
Right. Yes, absolutely. And it's it's sort of mind-boggling to me. Like already a transmissible tumor is crazy, right? Like as you said, when the devil facial tumor disease was identified, it was only the third documented transmissible tumor, and the other two are sort of benign. They're sort of like cancerous growths, but they're not actually that deadly in the populations
Right.
that they're in. So to have this one species have 2 out of the 4 known examples of a transmissible tumor is sort of remarkable and also just really, really sad.
Yeah, and it sort of gives you a sense of how damaging having a restricted MHC complement is.
Yeah.
Right? It's unlikely, it seems unlikely that it just happened to be that these 2 facial tumors, hit the Tasmanian devil population. It's much more likely that, unfortunately, these devil populations are so bottlenecked already, and their social interactions are so aggressive, and so they make contact with each other in ways that other species don't necessarily. It seems like there's this sort of perfect storm going on where they are both much more susceptible and much more susceptible to transmitting than other species.
Right. Yeah.
So, yeah, I think hopefully. So, they did talk a little bit. Apparently, there are vaccination efforts going on in these populations. They're hoping to try to get some sort of immune response against this thing, with the idea being that if you could convince what's left of the immune systems in the healthy animals that this tumor is foreign prior to their exposure, maybe you can prevent it, stave it off.
Yeah, I'm sort of shocked just as an aside, like the version 2, it sounds like is from a male.
Yeah.
And I'm sort of shocked that the Y antigens aren't sufficient to generate an immune response, at least in the female devils.
Yeah.
Um,
It
you'd
does
think that
make.
would limit the transmissibility, but
Yeah. So they talk about the karyotyping a little bit. I did some quick Wikipediaing on this.
it's a nice verb. I like that.
Yeah.
Wikipedia-ing.
Wikipediaing. So Tasmanian devils have 13 chromosomes. And it turns out that these cancers really have 9 functionally. They actually have 13 or 14, but really only 9 are actually recognizable as true chromosomes. And so I think that these cancers remind me of like HeLa cells, where they're just an absolute mess because they've been transported, animal to animal so many times. They're just a cellular wreck that seems to be passed well. So I think probably the Y antigen is the last thing that's coming up on this.
Yeah, that's fair. But it's also interesting to think about, like, um, one of the things we talk about often in epidemiology for like microbial infectious diseases, viruses and bacteria, is that you often have coevolution where the virus or the bacteria, the pathogenic agent, will also evolve to be, less virulent, less deadly, at the same time as the host is evolving to combat the disease, right? Because it's not necessarily beneficial if you are a pathogen that is so virulent that you kill your host before it's able to transmit, you are not a successful pathogen. And this cancer is now acting sort of like a pathogen. It's also, the tumor itself, the cancer, is also under selective pressure. And so I think another interesting question that's not addressed in this paper, but might be interesting to look at, is how the tumor is evolving
Yeah.
to potentially suppress the immune system or to be less virulent and less deadly to its host, because you can imagine a tumor that is growing more slowly is going to have more opportunities to transmit to additional devils, particularly as the population of devils crashes and there are less opportunities for transmission. You can imagine that the tumor that is surviving is likely to evolve to be less virulent, less deadly to its host, in the same way that a virus or a bacterium would evolve.
Yeah. Unfortunately, it's not doing a great job of it. And so they're all dying. So I think that we have a ways to go before the tumor equilibrates. It sort of reminds me of the Ebola infections in humans,
Mm-hmm.
right? So Ebola doesn't have a ton of contact with the human population. And when it emerges in a way that it's clear that humans and Ebola haven't had much interaction, right? Ebola just rips through the populations, it kills a whole bunch. So we heard a lot about that at Emory here in the CDC. We had a lot of the Ebola patients come back, and it turns out that you can give supportive care to these Ebola patients if you bring them out of Africa. And so what I would suggest is we start to pull these devils back to Emory CDC, since we had a pretty good response rate there. And we can also then say that the devils went down to Georgia. What do you think?
Except it's going to be devils coming up to Georgia
I
because they're coming from
know.
Australia.
So, yeah, but that's all a perspective, right? There's no
Like,
such
sure, if
thing
we
as
want to put
north south.
Australia on top, if we want
Okay.
to flip the globe, that was pretty bad. That was a
It
real
was terrible.
stretch. And I saw you reaching for it, and I feel like you spent days coming up with that.
I did. I was thinking about it the whole time that you were talking about coevolution. I was trying to get a Devil Went Down to Georgia reference in.
Uh-huh.
Yeah.
That was
I mean,
nice.
it could have been worse.
That's true.
It was a long
That's
lead
true.
up, though, for a low payout.
I mean, it's going to be the show title, but I'm not happy about it.
All right, good. Well, it's settled.
All right. So I feel like maybe Tasmanian devils are going to become a new theme on the podcast. We haven't talked about Cavendish in a while, which
true.
makes me sad.
Yeah.
I feel like we need to have these themes come back more regularly. But this has been Audiommunity. I think we're done, right?
Yeah, I think we're good.
Okay. So this has been Audiommunity, recorded on September 13th. I'm Kevin Bonham, and joined by Matt Woodruff. Kate is MIA, possibly buried with Jimmy Hoffa. We don't know.
Could be.
You can find us on Facebook and other places. I'm on Twitter and Google+. Matt is nowhere on social media, but the podcast is on Facebook. We are at emmunity.org, emmunity.org. Oh, you know what we should do is we should talk about the mail that we've gotten.
Oh, we have gotten a little.
So, and we have email that we should talk about. So the first is from Alex, who wrote to us back in June, and I was kind of delinquent and didn't respond until yesterday, which I feel bad about. I'm sorry, Alex. But Alex writes, "Hi, Immunity team. First of all, I'm a big fan of the podcast, but I've already mentioned that in some comments." I'm going to repeat. So Alex writes, "Hi, Immunity team. First of all, I'm a big fan of the podcast, but I've already mentioned that in some comments. So I will leave that for now, in parentheses, though it's a really cool podcast. I'm thinking about setting up an immunology podcast myself, but one already exists and it's really good. Thanks, Alex. And she would need pod partners and editing crew, so that might be a limitation." I think maybe that's a solicitation to invite Alex onto our podcast, which we should maybe think about.
Yep, we could consider that.
So she writes, "Anyway, I'm writing to you with a suggestion for an episode. It will be a pretty selfish suggestion, but let me explain. I'm fine with selfishness. I do this all the time. I'm an immunology major in the UK, going into my final year. Congratulations. And really, immunology, especially regulatory processes, tolerance, autoimmunity, is my thing. There is one thing, however, that I didn't consider choosing the field." I don't know if I read that correctly.
That I didn't consider when I was choosing the field. I think that
yes.
was the idea there.
Right. I'm terribly allergic to mice. I know it seems like a stupid choice then. So now I'm looking into it a bit more to see whether I have any chance of contributing something to the field if I cannot work on mice, especially that I will be applying to graduate school this year. So I looked particularly at organoids and organs on a chip technologies, and she mentions a couple of particular references, and I was wondering, what is your opinion on the direction of this research? I guess you're one of the people that are deep in the field that I have access to ask these questions. I was wondering whether you could talk a bit about whether the technology is promising and whether you know something about cool research that is ongoing which will utilize these systems. So organoids are super cool. These are basically, like, one example that I'm familiar with is intestinal organoids, where you basically take out portions of a mouse intestine, you loop it and connect it together, and then you essentially have a little mini in vitro intestine that you can then do cool things with. You can study microbiomes, you can study immune responses and stuff like this.
I actually had a collaborator that utilized those to look at antigen delivery from the lumen of the intestine into the parenchyma.
Oh, cool.
Yeah.
So super cool. Like any, you can also do human organoids. I think you can take like biopsies of human intestine and you can do cool things. I think like any model system, it's a model system and you have to be aware of the limitations. It's also worth considering that, for most of my PhD, I didn't work with mice. I think Kate is I think she's constitutionally opposed to mouse work. She refuses to look at mice. Matt's work obviously would not really be possible if you didn't work with mice, but you can definitely do it. It is definitely possible to do really cool immunology only working in cell culture or working with organoids.
Or working with humans. So Stanford's immunology program right now is essentially designing itself to be almost entirely human-based. I think that there's a lot of really good arguments for using mice to explore mechanistic questions about immunology. I think that they've yielded a lot of information that would have been possible otherwise. But the idea of translational immunology now is huge. I mean, the immuno-oncology field is almost exclusively focused on human biology. So, yeah, I think that there is a ton of opportunity to work in models other than mice and make a really significant contribution to the field. I work in vaccinology. I'm an immunologist that works on vaccines, but the entire vaccinology field still sees non-human primates as sort of the gold standard model for testing vaccines, so there's a lot of work being done there. And on top of that, it doesn't sound like you're a bioinformatician, but there's a ton of work to be done.
But you should be.
Totally, yeah. Kevin will argue that you should be. But there's a ton of work to be done in silico as well, and you won't get anywhere near a mouse. So I would certainly encourage you not to drop immunology because of an allergy to mice. I think that there are tons of ways that you could find interesting research that would be really important to the field.
And by the time you get to be a PI, I should say, one of the coolest PIs at Harvard, also one that spends the most money on mice of probably any faculty member at Harvard, is herself allergic to mice.
Who's allergic to mice?
Diane Mathis.
Is she really?
Yeah.
I didn't know that. That's a great fact. Yeah, she's a hardcore mouser too.
Yeah.
There's a lot of mouse work being done there.
Yeah. I think at one point Dimitri told me what the mouse bill in the CBDM lab was, and it was staggering. It's more than most labs have as their 10-year budgets is just spent on mice in those labs. So
yeah, no, it's
anyway,
a lot of mice.
Okay, so do you want to take the email from Michael? Do you have that up?
Yeah, let me find it real quick. Yeah, and so we have a second email from Michael. And Michael has a question about oral adjuvants and food allergies. So he writes, I've been kicking around the idea in my head for some time about the possible role of some type of adjuvant playing a role in food allergies. My naive view would be that it has to work. Wash down a couple of peanuts with some Freud's Complete and a little bit of LPS, and if you survive, voila, peanut allergy. However, given that aluminum hydroxide, or alum, as you may know it, is in many antacids, and aluminum is frankly everywhere, and we do not all become allergic to everything we eat, there must be some way of mitigating these reactions. Presumably, there are all flavors of immunology going on in the gut all the time, and on average, it does seem quite good at picking and choosing. Needless to say, immunology is dizzyingly complicated. I agree with that. So I must profess my profound gratitude for your podcast. I still don't understand it, but at least I have a reasonable idea of exactly what it is I don't understand, which is all of it. And I think that, Kevin, you and I would both agree that we're probably in the same boat there.
Yeah.
So
we have incrementally more knowledge than other people, but that is not saying a whole lot.
Yeah, exactly. So the basic question is, how does the gut distinguish between self and non-self? And as you sort of correctly surmised here, it's terribly complicated. You would think that by overloading the gut with danger signals and then putting in some foreign antigen, you would get a pretty nasty allergic response. And it turns out that at some level that is true, but at some level it's sort of not. So allergy is a funny thing because allergy often utilizes a different arm of the immune system than really ramps up when you're talking about the breaking of tolerance in the gut. So we've talked a little bit about the mouse colitis model on this podcast before, and the way the mouse colitis model works is you basically just feed mice detergent for a couple days. So it's a mild detergent, but it's enough where it basically strips away the mucosal layer in the intestine, and it does damage to the epithelium. And the result of that is this gigantic collective freak out of your immune system. And your immune system in the gut is extraordinarily highly regulated, and it works with the surrounding stroma. And when you have such a huge disruption to that normal anti-inflammatory barrier immunity environment, your immune system kind of goes nuts and it starts responding to all kinds of things that it's not supposed to respond to. And the result of that, rather than it being allergy, is basically a really good model for ulcerative colitis. You get a break in self-tolerance and you get antigenic spread like any other autoimmune disease. Basically, you get full-blown gut autoimmunity. So I think the answer to your question on the surface is, if you provided enough danger signals in the gut, I wouldn't expect so much an allergic reaction. I would expect more that you trigger some sort of huge inflammatory event that may or may not lead to autoimmunity.
I think it's worth pointing out that in a normal gut that isn't being fed detergent, your gut is already filled essentially with LPS and the equivalent of complete Freund's adjuvant, which is basically just emulsified mycobacterium.
Yep.
So your gut is filled with danger signals. The thing that SDS does is that, as you said, it disrupts that barrier immunity. So the immune system won't normally respond to things in their gut because things that are outside your epithelium, that is, they're in the lumen of the gut, are not actually dangerous. There's crap loads, forgive the pun, of bacteria that would be dangerous if they were in the lamina propria, behind the epithelial barrier, but in the gut, they're just fine. And as you mentioned, the bacterial triggers for the immune system are going to lead to different responses. And actually, one of the treatments for allergies that kind of works sometimes, but not always, is actually immunizing a patient with allergen. So you sort of do an injection of effectively aluminum hydroxide plus the allergen, and the idea there is that you're trying to shift the immune response towards type 1 instead of the typical allergic response, which is type 2, which is the antihistamine, mast cells exploding, and all that stuff. So yes, very complicated, but great question. And I think we've admitted on previous podcasts that allergies and type 2 immunity are like the redheaded stepchild of immunology and are woefully undercovered and understudied, I think. Because frankly, type 2 immune responses are to combat worms, and in the Western world, we don't get worms, and so they have not been a major focus of study. But super interesting and a great question.
Yeah, very cool.
So thank you for your comments. Please, other listeners, if you have comments, send them to us at, I think it's comments@emunity.org. Pretty sure there's also a form on the website, emunity.org, EMMUNITY.org. You can find us on Facebook, and I'm on Twitter. And the music at the beginning and the end of the show was composed by Rachel Reinick. And we'll see you next time.